Methiocarb is a carbamate pesticide which is used as a bird repellent, insecticide, acaricide and molluscicide since the 1960s. Carbamates are widely used in agriculture as insecticides and herbicides. They are preferred instead of organochlorines because organochlorines are long-lasting persistent in crops. Methiocarb has contact and stomach action on mites and neurotoxic effects on mollusks. Seeds treated with Methiocarb also affects birds. Like the other carbamates, Methiocarb inhibits reversibly acetylcholinesterase activity resulting in a cholinergic stimulation making Methiocarb a potent neurotoxin. It can even cause acute toxicity in humans if exposed for long periods of time or a sufficient dose. It is not listed as a carcinogen. Methiocarb is also a known poison to water organisms. Other names for Methiocarb are mesorol and mercaptodimethor. Structure and reactivity. The carbamate functional group in Methiocarb can be cleaved by cholinesterase to result in the carbamate, which binds to the cholinesterase, and the aromatic alcohol. Synthesis. Methiocarb 3 is synthesized by Bayer from 4-methylthio3, 5-xylenol-1, and methyl isocyanate 2. The xylenol-1 will act as the nucleophile in this reaction, attacking the partially positively charged carbon in the isocyanate 2. Mechanism of action. The product of the cleavage of the carbamate group of Methiocarb is methylcarbamic acid, which is bound to cholinesterase after the reaction. The normal function of cholinesterase is to cleave the acetyl-choline bond, which results in the binding of acetic acid to cholinesterase, which is a fast reversible reaction. The carbamic acid also reversibly binds, but the hydrolysis of the bond is slower and therefore the acid inhibits the function of cholinesterase, which results in elevated cholinesterase levels. In comparison, organophosphates inhibit irreversibly and will therefore inhibit the acetylcholinesterase even more. In addition to its cholinergic effects, Methiocarb has been found to be an endocrine disruptor, acting as an estrogen, antiandrogen, and aromatase inhibitor. Metabolism. Methiocarb is biotransformed in the liver mainly by sulfoxidation. This can happen to Methiocarb itself but also to the phenol group, which is cleaved from Methiocarb by cholinesterase. In some cases this same sulfur can be oxidized once more to give the sulfone. A minor pathway that occurs is the hydroxylation of the N-methyl. Absorption. Methiocarb can be taken up through different routes. The most common for humans is uptake through the skin or as an aerosol, because of its use as a pesticide in agriculture. For insects and birds this would be by the oral route. The NOAELs of these routes have been determined as follows. For the oral route, the NOAEL is set to 3.3 mg per kilogram per day for rats, based on a 2-year study. For absorption through the skin the NOAEL is set to 150 mg per kilogram per day for rabbits, based on the reduction of food consumption. When Methiocarb is fed to rats at a dose of 50 ppm, it gives a reduction of brain cholinesterase by 14% and 5% in males and females respectively. When Methiocarb is administered as an aerosol to rats, the highest concentration, 96 mg per cubic meter in solvent, showed signs of involuntary muscle contraction, tremors. These signs weren't observed in the other groups. The brain acetylcholinesterase is reduced in comparison to the solvent controls, to 61% and 74% for males and females respectively. There were no changes in organ weight. The NOAEL was determined to be 6 mg per cubic meter based on the reduction of brain acetylcholinesterase activity. Distribution. To determine the distribution of Methiocarb through the body, carbon-14, 14C, labeled Methiocarb studies have been performed on rats. About 8 hours after IP injection of 14C, Methiocarb more than 20 is present in the kidneys, 14 in the lungs, 14 in the heart, 6 in the body fat and 26 in the red blood cells. All the numbers are a measure of the radioactivity in dpm by 103 per grams of dried tissue. 30 minutes after treatment gave for all tissues except body fat, much higher values indicating that elimination takes place shortly after injection. Also, an increase in all tissues except the red blood cells has been observed between 2 and 4 hours after injection. This indicates that after 2 hours redistribution takes place shortly followed by elimination. This radioactivity study only measured the 14C, so the compound could already be metabolized to different compounds with different toxicities, which is not indicated in this study. Toxicity. Short-term toxicity. In rats, the cholinesterase activity fell down to 50% of the control values in 27 days, where the dose applied in their diet was 2 mg kg BW in the first 3 days and 4 mg per kg BW for the next 24 days. No abnormal clinical signs were observed. In rabbits, Methiocarb was applied to the skin to a group of 10 at doses of 0, 60, 150 or 375 mg per kg BW per day for 6H per day. 2 out of 10 rabbits with the low dose did not survive and with the high dose had a reduced food consumption. Cholinesterase activity was reduced in males with a high dose at 14 and 21 days of treatment. There were no intergroup differences observed in cholinesterase activity among females. The erythrocyte acetylcholinesterase activity is apparently not inhibited in a dose-related fashion. The duration of the study was 24 days. Long-term toxicity. In mice, a 1-year study of 50 males and 50 females was performed. The mice received diets containing Methiocarb at doses of 0, 15, 43 and 130 mg per kg BW per day in males and 0, 20, 57, and 170 mg per kg BW per day in females. Food consumption, behavior and mortality rate were not affected at any dose. At 1 month, the decrease in plasma acetylcholinesterase activity was the highest and the smallest reductions were observed at 24 months. Brain acetylcholinesterase activity was also lowered, more in males than in females. In rats, a 2-year study of 60 rats was performed. The rats received diets containing 0, 3.3, 9.3 and 29 mg per kg BW per day for males and 0, 5, 14, and 42 mg per kg BW per day for females. Food consumption, behavior and mortality rate were not affected at any dose. The total protein concentrations were raised at higher doses of Methiocarb. The plasma acetylcholinesterase activity was lowered at the high dose at day 1 and from 8 weeks onwards in males and at day 1 and 1, 2, 4 and 13 weeks in females. No brain acetylcholinesterase activity was observed. Environmental toxicity. Because Methiocarb is widely used as an insecticide on crops, environmental risks were also studied to establish safety risks for human health. The metabolism of Methiocarb in plants, soil and water have been proposed from radiolabeled 14C Methiocarb studies. In plants, the major metabolites were Methiocarb sulfoxide and Methiocarb sulfoxide phenol. Environmental fate in water and soil has been determined from the metabolites formed by an aerobic degradation, photolysis, adsorption and leaching of Methiocarb. In soil, the half-life of Methiocarb sulfone phenol is 20 days, Methiocarb sulfoxide phenol is 2 days, Methiocarb 1.5 days and Methiocarb sulfoxide 6 days. Methiocarb is mainly metabolized to Methiocarb phenol and minor to Methiocarb sulfoxide and Methiocarb sulfoxide phenol. Also, after 217 days, no Methiocarb or metabolites are present anymore in the soil. This is because a lot of gets metabolized to CO2. In water, no Methiocarb was present already after 32 days. The half-life of Methiocarb in water is strongly pH dependent, but at pH 7 the half-life is about 28 days. Efficacy. Methiocarb is used as toxin for different purposes. It ranges from snails, insects, rodents and even as a bird repellent. As an insecticide it is effective for thrips and has a low dose that is lethal for these animals. The LC99 for suspension concentrate is 0.34 grams per liter and for the wettable powder it is 2.30, which is a bit too much for effective use. 0.10 for the use as a molluscicide. Methiocarb is effective, but at a high dose. In a research with E. vermiculata, Methiocarb showed to be the most effective as topical applicant, although DMSO was used as a solvent. The LD50 is 414 micrograms per snail and the LD99 is roughly estimated 1,400 micrograms per snail for Methiocarb. In comparison to methimyl, which was more effective, with its LD50 was 90 micrograms per snail, which is a lot lower than the LD50 of Methiocarb, as snail-bait Methiocarb has the same effectiveness as methimyl for 1% mass percent and 2%, but the LC50 of Methiocarb is higher than the one of methimyl. 0.93, 0.31. They both reached an average mortality of 85%, by the use of 2% Methiocarb, methimyl bait, in another comparison study, with M. obstructa, between Methiocarb and methimyl. Methimyl showed again to be more effective. The LD50 in this study were 12 micrograms per snail for methimyl and 27 micrograms per snail for Methiocarb. These compounds were topically applied on the snails and these compounds were first dissolved in 95% ethanol and diluted with water to make the concentrations. As an avian repelled to protect fruit, Methiocarb was in one research not effective. The birds still damaged the figs. This happened because the Methiocarb was sprayed on the fruit. The birds pinched the fruits or peeled the skin of the fruit and ate the meat of the figs. In that manner these birds are very little or not exposed to the repellent. In another study with Quelea, it was investigated if Methiocarb had an adverse effect on the food choice. It showed that when Quelea ate seeds with Methiocarb, the next time they would choose some other food. This shows that Methiocarb can be effective as a bird repellent. In one study Methiocarb is shown to be not very effective against mice as a rodenticide. In the first field trial, snail pellets of Methiocarb were spread across the land and killed almost 23% of the initial mice population in one night, but the population did not decrease, probably because of re-invasion of the neighboring land. There hasn't been searched for carcasses after that, but birds were seen scavenging on carcasses. In the second field trial, grain was covered in Methiocarb and strychnine and it showed a mortality rate of 40% for Methiocarb and 90% for strychnine. Although Methiocarb seems to be effective at first, mice develop an aversion for the Methiocarb, which makes it not very effective as rodenticide. Suicidal poisoning. Methiocarb is a plant protection agent and while suicide with these type of toxins is rare, there is one case reported of a suicide with Methiocarb. An 80-year-old woman in Germany killed herself by drinking a bottle of meserol. The red-pink fluid was on her clothes, face, and hands, probably because of the vomiting, and in the gastrointestinal tract as in the respiratory tract. The toxicological examination showed that the Methiocarb uptake wasn't completed and the concentration of Methiocarb and its metabolite in the urine was low. This is due to the short duration of exposure. Elevated concentration of Methiocarb may be the result of post-mortem uptake, but it could also be the post-mortem redistribution of Methiocarb and metabolites from the gastrointestinal tract. The conclusion of the toxicological examination was death by acute poisoning of Methiocarb. The amount of Methiocarb in the stomach is calculated to compare it with the LD50 of rats. The amount of Methiocarb is estimated to have been 6.1 gram by a stomach volume of 1 L. The weight of this woman was 53 kg. That would make 115 mg per kilogram BW. When compared to the LD50 for rats, which is 30 mg per kilogram, it is reasonable to say that this woman died of poisoning from Methiocarb. Bear in mind that this is only the amount of Methiocarb found in the stomach and that the rest was already Methiocarb distributed through the body. Semi-quantitative analysis was performed by the approximation of similar extinction coefficients of mercaptodimethor and its metabolite discarbamoyl mercaptodimethor at wavelength 200 nanometers. See also: Methiocarb in the Pesticide Properties Database (PPDB). References